IB Bio Human Physiology covers 7 body systems: digestion, circulation, immune, gas exchange, nervous, endocrine, and reproductive. This will be the last movie in which I list them all, as I've just done. Now, part two, this movie is the second of 2 parts on the digestive system. This movie includes the details surrounding the absorption of nutrients from digested food into the bloodstream. The essential idea is the structure of the wall of the small intestine allows it to move, digest, and absorb food. Here is the first of 2 slides that provides an outline of all parts of Topic 6, Human Physiology, for SL and HL students. Use the outline to find the movie you need for review. This movie is focused here. Here is the second slide of the outline for all parts of Topic 6, Human Physiology, for both SL and HL students. Before moving forward, let's review the relative positions of the organs of the digestive system. We have the mouth, the esophagus, the stomach, small intestine, the large intestine, and the anus. The pancreas, seen here, and the liver are associated with digestion. Ducts bring their secretions to the small intestine right here, just below the stomach, into the small intestine. The gallbladder, seen here, stores bile, which is manufactured in the liver. Here's a chance to review the names and relative positions of all the digestive system organs. You can turn the movie off now or follow along with me. A is the esophagus, B is the stomach, C is the pancreas, and here is the duct that takes the pancreatic enzymes into the small intestine, which is D. E is the large intestine, F is the liver, and G is the gallbladder. In moving forward, keep your eye on the function of each structure, shown here. The liver, seen here, and the pancreas, seen here, both secrete digestive juices into the small intestine. The liver secretes bile into the small intestine, and the bile emulsifies fats. Bile does not contain enzymes. Now, the pancreas secretes enzymes into the lumen of the small intestine. Enzymes digest most macromolecules into monomers in the small intestine. The glands that manufacture and secrete enzymes into the digestive tract are the salivary glands, the stomach, the pancreas, and the small intestine itself. The liver releases bile to the small intestine. Bile emulsifies fats, which is a physical breakdown, not a chemical breakdown. Our focus in this movie will be on the pancreas and the small intestine, and here are the 2 relevant IB syllabus statements related to the pancreas. The pancreas secretes enzymes into the lumen of the small intestine. State that amylase, lipase, and an endopeptidase are secreted by the pancreas into the small intestine. The endopeptidase is an enzyme that breaks peptide bonds toward the middle of the larger polypeptide. The pancreas produces pancreatic juice that has a pH of 7 to 7.5. Pancreatic juice has the following enzymes: pancreatic amylase, pancreatic lipase, and pancreatic endopeptidases. Endopeptidases break peptide bonds in the middle of the larger polypeptide molecule. Trypsin and chymotrypsin are examples of endopeptidases. Amylose, starch, is hydrolyzed by the enzyme amylase into maltose molecules. This should be review for you. Maltose is hydrolyzed to glucose by the enzyme maltase. The enzyme maltase is synthesized by glands in the small intestine. Other enzymes synthesized in the small intestine include sucrase and lactase. Sucrase hydrolyzes sucrose, and lactase hydrolyzes lactose. The hydrolysis of these disaccharides into monosaccharides should be reviewed from our study of biochemistry. Remember, you need to know the 3 disaccharides that I've mentioned here: maltose, sucrose, and lactose. In this slide, we have a polypeptide, and endopeptidases break the polypeptide in the middle of the molecule, producing peptide fragments. Exopeptidases remove amino acids from the ends of the molecule, here or here. Exopeptidases break polypeptides into individual amino acids. Pancreatic cells, seen here, and here, 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 and here, are clustered around ducts. These pancreatic cells are clustered around a duct that leads to the small intestine. Now, all the cells are clustered around the duct so that they can empty their enzymes into the duct by exocytosis. You can see the Golgi vesicles preparing to release the enzymes into the duct leading to the small intestine. The clustering of cells around a duct is called an acinus, and it provides a wonderful example of a form-function relationship. Remember, the duct takes enzymes to the lumen of the small intestine. Here is another slide displaying acini, which is plural for acinus. On the left, a drawing, you can see the clustering of pancreatic cells around the duct. Each cell can manufacture enzymes and move those enzymes into the duct by exocytosis. On the right, you can see micrographs displaying pancreatic cells clustered around a duct. Here is another micrograph of a pancreatic acinus, cells clustered in a circular fashion around a duct. Cells such as this one here has Golgi vesicles preparing to release enzymes into the duct by exocytosis. Ribosomes, seen here along the ER, manufacture the protein enzymes of a pancreatic cell, seen here. The proteins are synthesized from amino acids that are available in our diet. Then vesicles from the ER move to the Golgi, and then Golgi vesicles carry the digestive enzyme toward the cell membrane for exocytosis into the lumen of the duct. Amylases, lipases, and endopeptidases flow from the ducts in the pancreas into the small intestine for the digestion of macromolecules. You've seen these IB syllabus statements before. They're here just to remind you of their presence. Starch, glycogen, lipids, and nucleic acids are digested into monomers. Cellulose remains undigested. Enzymes digest most macromolecules in food into monomers in the small intestine. In this micrograph, we have a cross-section of the small intestine. Now, this is the small intestine, not the pancreas. The small intestine is also an exocrine gland, manufacturing enzymes involved in digestion. The glands are at the base of these finger-like projections called villi. In this diagram of the small intestine, you can see the circular and longitudinal muscle layers that antagonistically contract to mix food and enzymes together and move food along the digestive tract. You can see the folds in the wall of the small intestine, and you can see the villi, which are finger-like projections that extend into the lumen of the small intestine from those folds. Here is a close-up of a villus. The cells that line the villus make up the epithelium of the small intestine, and nutrients must pass across those cells to reach the bloodstream. The long, slender shape of the villus, as well as cellular extensions called microvilli, increase the surface area over which nutrients pass into the bloodstream. In this diagram of the villi of the small intestine, you can see the muscle layers, the circular muscles and the longitudinal muscles that antagonistically contract to mix enzymes in food and move the food along the digestive tract. The mucosa, as it is called, includes the glands that secrete enzymes, includes the villi, seen here, and the epithelium composed of the cells that line each villus. Here is another image of villi. Muscle layers are located here. The mucosa includes the glands at the base of the villi and the villi themselves. The epithelium is composed of the cells that line each villus. Blood vessels penetrate each villus, allowing for the absorption of monomers from the lumen of the small intestine into the bloodstream. Here are two relevant IB syllabus statements. Identify the tissue layers in a transverse section of the small intestine. Explain how the structure of the villus is related to its role in absorption and transport of the products of digestion. Here is a clear diagram of one villus. The epithelium can be seen clearly. Blood vessels penetrate the villus. Villi increase the surface area of epithelium over which absorption is carried out. Villi absorb monomers formed by digestion, as well as mineral ions and vitamins. So the functions of the small intestine are digestion and absorption. For absorption, the small intestine provides a beautiful example of a form-function relationship with a high surface area. The high surface area of the small intestine comes from its long length, the folds in the wall, the villi, and the cellular extensions along the epithelial layer known as microvilli. As well, the villi bring the monomers in the lumen of the small intestine into close proximity with the bloodstream within each villus. Villi increase the surface area of the epithelium over which absorption is carried out. Capillaries are located here within the center of each villus that serves to transport those nutrients from the intestine to the interior of the body. Epithelial cells in the small intestine have membrane-bound extensions called microvilli, seen here. The membrane goes out and around each one of these cellular extensions. And the epithelial membrane has membrane-bound enzymes, which further enhances the digestion of polymers. Villi absorb monomers formed by digestion, as well as mineral ions and vitamins, because of the high surface area and the close proximity to the bloodstream. And as the monomers of digestion, as well as mineral ions and vitamins, move into the bloodstream, the blood moves to the liver for processing of nutrients. Digestion of macromolecules occurs in the small intestine. Then the monomers, mineral ions, and vitamins are absorbed into the bloodstream. Then the monomers and the mineral ions and vitamins are taken up by cells and assimilated into larger molecules or metabolic processes. And that brings us to the last IB syllabus statements for this movie. Describe the processes occurring in the small intestine that result in the transport of products of digestion to the liver. Describe the different methods of membrane transport required to absorb different nutrients. The total surface area of the small intestine facilitates absorption. High surface area arises from the long length of the intestine, folds in the wall, villi, and microvilli, all of which can be seen in this image. It is across the epithelial cell membrane that monomers such as glucose, amino acids, fatty acids, glycerol, and nucleotides are moved into the bloodstream. Microvilli, seen here, are extensions of the epithelial cell membrane, increasing the surface area for the absorption of monomers. Absorption is an important term. It means diffusion, facilitated diffusion, active transport, and penocytosis. Absorption occurs across the epithelial membrane, seen as the microvilli in this micrograph. Absorption is a word with meaning. Absorption could be diffusion, or facilitated diffusion, or active transport, or penocytosis. Glucose, amino acids, and nucleotides are transported into the bloodstream by active transport. Water and fatty acids move passively into the bloodstream by diffusion or facilitated diffusion. The blood leaves the digestive tract and moves to the liver. Diffusion, facilitated diffusion, and active transport as processes serve to move monomers from the lumen of the digestive tract into the bloodstream that is flowing within a villus. Diffusion, facilitated diffusion, and active transport should be review for you. If you need to refresh your understanding of these processes, stop the movie and study this slide. Glucose and amino acids are actively transported across the epithelial cells of the villus to reach the bloodstream. The active transport of glucose and amino acids is a co-transport system involving sodium. A co-transport system involving sodium. Both molecules are facilitated across the epithelial cell membrane along with sodium. Sodium is actively pumped out of the cell to be available for the co-transport of glucose or amino acids. In this diagram of an epithelial cell of a villus with its microvilli, we can see the co-transport of glucose alongside sodium happening right here. Sodium is actively pumped out of the cell here. Fatty acids and monoglycerides passively move out of the lumen of the digestive tract and into the epithelial cell, but triglycerides are reassembled in the epithelial cell, then moved into the lacteal of the lymph system. Fatty acids and glycerol, after leaving the digestive tract, are reassembled into triglycerides in the lymph vessel, which runs through the center of the villus. This lymph vessel is also known as the lacteal. Fluids in the lymph are moved to the circulatory system. Endocytosis is also a mechanism by which epithelial cells of the villus take in monomers, mineral ions, and vitamins. When fluids are taken in by endocytosis, we use the term penocytosis. Here is a diagram of one epithelial cell that lines a villus. You can see the extensions of the cell membrane called microvilli. You can see the extensive rough ER, evidence of the cell's activity in manufacturing enzymes, which can be seen being moved into the lumen of the small intestine by exocytosis. Numerous mitochondria serve to provide ATP to drive active transport, actively bringing in monomers from the lumen of the intestine, monomers like glucose or amino acids. You can see penocytotic vesicles transporting the products of digestion into the cell, and tight junctions, seen here and here, are membrane proteins that bind the membranes of two adjacent cells, bind those cells tightly together. Tight junctions would be another role of membrane proteins. You can study this slide on your own. I've identified the structures that I spoke about in the last slide. So the villi and the microvilli provide immense surface area for the absorption of the products of digestion, and the epithelial cells of each villus are engaged in diffusion, facilitated diffusion, active transport, and endocytosis, penocytosis, as they move monomers, mineral ions, and vitamins toward the bloodstream. And once those nutrients get to the bloodstream, the circulatory system moves them to the liver for processing. And that brings us to the end of IB Bio Human Physiology Part 2, Digestion, Absorption, B.